Okay, welcome back everyone. We're continuing with Chapter 3, the cellular level of organization. So this kind of goes back to what we were talking about in the previous chapter about how the smallest functional unit of life is the cell, and essentially that is the smallest area into which you can cram everything that's necessary to perform all the functions of life. Cells are organized into roughly three groups. Well, when I say roughly, I mean fundamentally. You have prokaryotic cells, eukaryotic cells, and archaean cells. Archaea are kind of funky because they have some traits from prokaryotic, some traits from eukaryotic. They're just they don't fit into this nice defined group, so we stick them over there. Prokaryotics lack membrane enclosed structures, eukaryotics possess them. Um, and this, of course, refers back to our domains. The word cell originated in the 17th century when Anton von Leeuwenhoek invented the first microscope and observed living cells. Robert Hooke coined the term cell when he was looking at slices of cork because the little divisions of the cork reminded him of the cells at a monastery where monks lived. And it just kind of stuck. So I'm not going to pronounce these properly, but I will give it a shot. Matthias Schleiden and Theodor Schwann came up with cell theory, essentially that all living organisms are composed of cells, whether they be unicellular or multicellular, that the cell is the basic unit of life, and that cells arise from pre-existing cells. And there was actually a period in time where everyone kind of thought that life just spontaneously arose from, literally from the ether is what they called it. And if you left a steak out, that the maggots would form from the ether in it, and that's just kind of how things worked. But then eventually people proved that, well, no, it was actually flies transmitting the eggs, and if you kept something in a sterile environment, nothing would grow in it, and so on and so forth. So, which brings us to our modern cell theory. Now, with that in mind, prokaryotic cells, they're in the domain bacteria and archaea. They lack a membrane-bound nucleus. They're usually unicellular, and they can be in chains or clusters as well as just a single cell kind of floating around doing its own thing. Some cause diseases, some are beneficial. Most are neutral. They don't really affect us one way or the other, except kind of outcompeting more pathogenic bacteria. So in that sense, they're indirectly useful. Prokaryotes are typically structurally simple, metabolically diverse, and adapted to extreme environments, which is why their metabolism is so diverse. You have to have a completely different way of gathering energy if you're living off of volcanic vents and using sulfur as an electron carrier at the bottom of the ocean than if you are living, say, in a hot spring, or not a hot spring, but a salt bed or something. They're just very different environments. Not shown here in this picture is thylakoids, but that's we'll get into those later. It's a flattened disc that contains pigment used by cyanobacteria and also other photosynthetic organisms to capture light so they can perform photosynthesis. The main thing here is you've got your fimbriae, which are just little hair-like bristles that allow them to stick to surfaces, flagella for movement. The capsule is outer coating outside the cell wall. Then you have the cell wall, plasma membrane, nucleoid, which is not a true nucleus, because again, they don't have a nice membrane-bound nucleus, but they kind of have the chromosomes sort of clustered somewhere, and that's what we call the nucleoid. And then you can see kind of speckled throughout here little ribosomes for protein synthesis. Eukaryotic cells are more complex, and they do have a nucleus and possess membrane-bound organelles. And this is all of your plants, animals, fungus, and protists. And again, protista is kind of one of those grab bag categories. If it doesn't really fit in either of the other or any of the other three, you just throw it over there. Okay, so plasma membrane. All cells are prokaryotic and eukaryotic are surrounded by a plasma membrane. This regulates the flow of things in and out of the cell, basically keeping the things in you want in and the things out you want out. And that's very important in particular cells for setting up like electrical potentials, so you can contract your muscles or fire a neuron or something like that. But in general, if you don't have a membrane in place here to maintain homeostasis, you cannot control the concentration of chemical reactions necessary to keep the cell functioning, and they die. So inside the membrane here is the cytoplasm, which is a semi-fluid medium. You got a lot of water, salts, dissolved organic molecules, just kind of floating around there. It kind of acts as a solvent for things to function in. Some eukaryotic cells have cell walls, which are typically plants and fungi, and some insects also have chitin in their exoskeletons, which is kind of a cell wall. But animals proper do not have cell walls. The chitin is just the chitinous exoskeleton is kind of a cell wall, but not in the sense that it's like part of the cell itself, like you see over here with the plant cells. Organelles refers to any well-defined structure that performs a particular function, and they like to use this analogy of a cell being a factory. You send raw materials in, they go to different organelles and they're processed properly, and then you get products out of them, and then you also have to pump waste outside the cell. So the nucleus stores the DNA. That's where all the information that governs how the cell functions is located. It's kind of like the master instruction manual. Within the nucleus, there's the nucleolus, where ribosomal RNA is made, and we'll talk more about that later. The nuclear envelope is a double membrane that separates the nucleus from the cytoplasm. It's kind of like another plasma membrane, further encapsulating the nucleus. And then you have inside that membrane nuclear pores, which allow the transport of proteins to and from the nucleus, and ribosomal subunits for protein synthesis. The ribosomes are where proteins are synthesized. They have a small and a large subunit, and we'll talk more about what each of those subunits is later. And then you have groups of, or clusters of ribosomes called polyribosomes. Poly means many, so many ribosomes in one place. Call it a polyribosome because biologists are creative with names. Some float around free in the cytoplasm, others are bound to the endoplasmic reticulum. Not all endoplasmic reticulum has ribosomes on it. If it has a ribosome on it, it's referred to as rough ER. If not, it's smooth ER. And the rough ERs usually helps process and modifying proteins. The smooth ER synthesizes phospholipids and steroids. Also stores calcium ions. You'll hear this referred to as the sarcoplasmic reticulum in muscle cells, but that's the same thing. It's just a different name because it's in a different cell type. The endomembrane system acts as transportation and processing units of the cells. They compartmentalize the cells so specific enzymes can be catalyzed in specific places, which is important because there's only certain things you want happening at certain times and in a certain order in order to maintain homeostasis and keep living. Part of the system, you have the nuclear envelope, the endoplasmic reticulum, Golgi apparatus, and multiple vesicles. And the vesicle is just like a tiny little membranous sac that just stores things for the time being. Golgi apparatus is the shipping center. You collect, sort, package, and distribute materials throughout the cells, or send them outside the cell to be excreted into the body, or into the body if you're talking about a multicellular organism. But if you're talking unicellular, it's just shipping out the waste. Lysosomes are membrane enclosed vesicles formed by the Golgi. They're essentially the garbage disposal. They contain hydrolytic digestive enzymes, and basically anything that's foreign or has broken down and doesn't work anymore, you send it to a lysosome to be destroyed and, if possible, recycled. Vacuoles, large membranous sacs, much larger than a vesicle, more prominent in plants. That's this big jello looking thing right here in the center. They provide support and are typically used to store water or pigments or toxins until they can be processed or used. Peroxisome is similar to a lysosome, except it uses uses peroxide to break down and catalyze things that way. So it's a little different mechanism for breaking things down, typically used for fats or biles, and in plants they oxidize fatty acids. Chloroplasts and mitochondria are essentially, well, the mitochondria is always referred to as the powerhouse of the cell because it undergoes cellular respiration. The chloroplast essentially does the opposite of that, and it uses solar energy to synthesize carbohydrates, which can later then be sent to the mitochondria to be broken down to produce ATP. So when later on you'll see the reactions for these, and they are exactly inverse. The exception being that the mitochondria doesn't produce light at the end of it. I mean, it kind of does because it produces heat, which is a form of light, but that's not really what we're talking about here. So chloroplast side of photosynthesis. The stroma is filled with a space-filled membrane that contains circular DNA and ribosomes. And then the grana is the stack of thylakoids, which we talked a little bit about earlier, and that's basically where sunlight's collected. Mitochondria is the site of cellular respiration. Double membrane again, matrix is the inner fluid space, and then they have these little cristae, which increase the surface area, and they'll also contain their own DNA. And we'll talk a little bit more about why they have their own DNA in just a minute. The cytoskeleton provides a scaffolding for the plasma membrane structures. You can also use projections of it into the intestine, kind of like microvilli, to increase surface area. They can form pseudopods in amoebas to allow them to move, and they can interact with motor molecules for movement, such as in your muscle cells. It's composed of actin, intermediate filaments, and microtubules. And the actin is two long, flexible, twisted chains, kind of like you see here. Intermediate filaments, as the name implies, are intermediate size between microtubules and actin, and they function to support the nuclear envelope, help form cell junctions, strengthen your hair, that sort of thing. Microtubules are assembled by the microtubule organizing center, and the most important is the centrosome, which is used to organize your chromosomes during cellular replication. When this malfunctions, you can get a daughter cell that is missing or has extra copies of DNA. Usually that will lead to the termination of that cell, but it also helps to maintain the shape of the cell and interact with motor molecules to cause movement, and forms the spindle apparatus during cell division. Centrioles are found in the centrosomes of animals, may be involved in microtubule assembly and disassembly. That's our best guess at this time. It's not we don't know completely or with complete certainty what they do, but that's the best guess at the moment. Cilia and flagella, hair-like projections composed of microtubules that aid in movement. In eukaryotes, cilia are much shorter than flagella. About the only place that we have flagella is on sperm cells. A good example of cilia, though, would be in your lungs. They kind of beat in unison and create a flow of mucus coming up out of the lungs, and the purpose of that is to clear any dust or debris you might have inhaled throughout the day, so that way your lungs don't become filled with gunk. When you smoke, it actually paralyzes the cilia, which is part of the reason why smokers tend to cough frequently. That and the smoke itself is an irritant. Also, cigarette smoke inhibits a inhibitor of an enzyme called alpha-1 antitrypsin, and alpha-1 antitrypsin will break down the connective tissue in your lungs and will cause the alveoli to collapse and result in pleural cavities being formed in your lungs, which will cause emphysema. So I recommend avoiding tobacco if at all possible. So let's see if I can zoom in on this without breaking it. So this is what we were talking about before here with the mitochondria and chloroplasts having their own DNA. At one point in time, you had this early prokaryotic cell that surrounded its nucleus with a membrane and allowed its functions to be separate from the rest of the cell. And it tried to eat a anaerobic bacteria, and that anaerobic bacteria essentially became an intracellular parasite. And unlike most parasites, it developed a symbiotic relationship, and that line went on to become animal cells as we know them today. And some of them did the same thing again with a photosynthetic bacterium, and that line went on to become plants. So this whole mess is referred to as the endosymbiosis theory. That concludes chapter 3. We've got one more chapter with the first exam's material, and then we'll have our first exam after chapter 4. If you have any questions, comments, concerns, feel free to send me an email, and I'll catch you in the next lecture.